Hello guys, today you will learn about the enzymes, which is the heart of the biochemistry. So what is the enzyme? Enzyme is referred to as biological catalyst. Biological catalyst. What is catalyst? Catalyst is the agent that increases or that changes the rate of the reaction. So this enzyme in cells, it is serves as catalyst. So the reactions are catalyzed by using enzymes. Like in laboratory we use acids and bases. Here in cells we use the enzymes. These enzymes, for example, why do we use them? For example, hydrolysis of the lipids requires water, but lipid is non-polar molecule, so it is not hydrolyzed easily by water. So additionally, we want what? We want enzyme called lipase, so that this enzyme helps they coexist with the water, and so many cellular reactions occur by using the enzymes. Why? Cell has around 60 to 70% is water. So the molecules which are found in cells are mostly the non-polar molecules. They cannot be reacted in the water environment of the cell. So enzyme is needed to enhance this reaction. So let's go into our today's discussion. I think it is clear why enzyme serves as catalyst. Any reactions which takes place in the cell are undergo reaction with the help of the enzyme. This enhances the rate of the reaction. Okay, let's continue. Enzymes are molecules that increase the rate of chemical reactions in living systems without being changed themselves. They are not changed. They can increase the rate of the reaction. They can bind to the substrates and can help substrates to be converted into the product. But the enzyme itself is not changed. That means does not increase, does not be used up. So this is one characteristic of the catalyst. Catalyst is that a substance that increases the rate of reaction without being changed or altered or without being used up. Okay, all enzymes are proteins except RNA enzymes, ribozymes. Enzymes can exist as simple or hollow enzyme. Simple enzyme is this enzyme that has only protein component, which is formed only from protein component. But hollow enzyme is formed from protein and non-protein component. Protein part of the hollow enzyme is called apoprotein, whereas the non-protein part of the hollow enzyme is the cofactors. So example of simple enzyme is pancreatic ribonuclease is one example of simple enzyme which is formed only from protein molecule. The other is hollow enzyme, complete catalytically active enzyme. Complete catalytically active enzyme. The protein part only inactive by itself, but the presence of the cofactor activates and the hollow enzyme becomes catalytically active. Okay. I told you protein component is called apoenzyme, non-protein component is called cofactor. Right? Now, to form the hollow enzyme, there is protein component, there is non-protein component. The protein component is called the apoenzyme, whereas the non-protein component is called cofactor. This cofactor also divided into two. Those are coenzymes and mineral ions, right? Coenzymes and mineral ions. That means when we say mineral ions, they help in the metallozymes. In metallozymes, the apoprotein part bound to the mineral ions tightly, and so they become active enzyme. When we talk about the coenzyme, it is formed from the it is the cofactor which is formed from the vitamins. So, for example, you can riboflavin, biotin, these all are the coenzymes. Okay, The protein part of the enzyme gives the necessary three-dimensional infrastructure for chemical reaction to occur. But the group is transferred from or accepted by the coenzyme. This coenzyme, for example, holoenzyme has two parts, that is apoprotein and a cofactor, which is a coenzyme. The apoprotein part forms the three-dimensional infrastructure for the chemical reaction to occur, whereas the coenzyme part either takes the group from that substrate or gives the group for that substrate. For example, some that is adenosyl methionine, this gives the methyl group. If you take PLP, this removes what? The amino group. So also fat plus takes hydrogen. Fat gives hydrogen. So this is how the holoenzyme works. Cofactors are heat stable, which can be divided into organic biomolecules, coenzymes, inorganic part as metal ions. Coenzymes can be co-substrate, which is transiently attached with the enzyme. For example, paradoxical phosphate co-substrate for amino transferase. This is the co-substrate. When we say this is the coenzyme which binds with the enzyme loosely or transiently. That means no damage occurs when this group leave or come. But the other cofactor or the other coenzyme is called prosthetic group. This prosthetic group permanently bound with the enzyme. If this prosthetic group is removed from the enzyme will be damaged. It is impossible to remove the prosthetic group from the enzyme without damaging the enzyme. For example, fat, biotin or vitamin B7 is prosthetic group for carboxylase enzyme, right? So if we remove the biotin from the carboxylase enzyme, the carboxylase enzyme's function will be altered, or it will be what? Damaged. Generally, coenzyme is essential for biological activity of the enzyme. Usually they are vitamins. They are heat stable. Okay. This is coenzyme, especially the B vitamins with coenzyme. For example, these are vitamins, right? These are vitamins. All of these are vitamins. These are coenzyme which are formed from them. These all are fat, also FMN, NAD, NADP. All of these are the coenzymes which are formed by the vitamin B. Okay, as you can see, this is the structure of the diagrammatical. This is the diagrammatical presentation of holoenzyme. This is apoenzyme, which is the protein portion of the enzyme that is inactive. This is coenzyme. So this coenzyme is the activator. When this goes and binds to this site, then this apoprotein forms three-dimensional structure for this substrate. This receives or gives group for the for this substrate. Okay, let's go into the metalloenzymes. As I have already said, metalloenzymes are the enzymes that require metal ions for their activity, right? Ions tightly bound with the enzyme. Okay, metalloenzymes, they require what? Metal ions for their activity. Ions tightly bound with enzyme, right? Tightly. When we talk about metalloenzymes, ions are tightly bound with enzyme. So if we remove this group, the enzyme will be damaged. For example, carbonic anhydrase needs the zinc ion, tyrosinase needs iron ion, also catalase needs the copper ion. This is all about the metalloenzymes. This uses metal ions. These metal ions are for the activity of this holoenzyme. It is necessary, right? If this is lost, the enzyme does not function properly. Okay. Also, there is other enzyme which is called metal activated enzyme. The metal activated enzyme is the enzyme which requires metal activated enzyme is the enzyme which is by itself it is active, but when we add the metal ions their activity will be enhanced. Okay, example, enzymes those need sodium, potassium, magnesium, zinc. These are metal activated enzymes. Hexokinase, phosphofructokinase, glucose 6 phosphatase. These enzymes are metal activated enzymes. Okay, what are the functions of the metal ions? Metal ions, by binding to the substrates to orient them properly for reaction, binding to the negative charges on the substrate, and orientation for the substrate. Okay, properties of the enzyme. Properties of the enzyme. Active site, it is a region on enzyme molecule that binds the substrate and converts it into the product. This active site has specificity. Active site is a small part of the total volume of the enzyme. It is a pocket on the surface of the enzyme. Binding of the substrate to active site of the enzyme forms enzyme substrate complex. Right? This active site, when you say it forms the three-dimensional structure for the substrate to bind to it. It is specific. When you say specific, it is maybe bond specific, stereo specific. Also, it has many specificity. The other is catalytic efficiency. In the presence of the enzyme, the rate of reaction can be increased by up to 10 to the power of 6-fold. The other characteristic is regulation. The enzyme can be activated or inactivated. That means, for example, glycogen synthesis activated by insulin. This glycogen synthesis also inhibited by glucagon. So this is the characteristics of the enzyme. It can be activated, can be inhibited. In the presence of metal ions, it can be chemicals, it can be hormones, neurotransmitters. Depending on this, they can be regulated. That means can be up regulated or down regulated. Enzyme specificity. I have told you in active site portion, in this the active site has specificity for the substrate. I said the specificity is of 3 different types. Those are stereochemical specificity, reaction specificity, and substrate specificity. Stereospecificity, when you say the enzyme acts on particular optical isomer. For example, the enzyme which acts on the L-amino acid oxidase acts on L-amino acids only. It does not work on G isomer, right? This is the one property of the enzyme. Substrate specificity. This means enzymes are highly specific for the substrates. This substrate specificity can be broad specificity or absolute specificity. When we say broad specificity, it has the specificity for the related molecules. For example, hexokinase works on all hexose sugars. But when we say absolute substrate specificity, this works only on one. For example, glucokinase, it phosphorylates only glucose, not fructose. This is all about the substrate specificity. Bond specificity. Enzyme also has, the active site of the enzyme also has the specificity for the bond. For example, some enzymes act on the peptide bond, that is called peptidase. Some enzymes act on the ester bond, those are called esterases. Some enzymes act on glycosidic bond, which is called glycosidases. So what we learn from this, the enzyme has specificity for the bond. So each bond has its own enzyme in the chemical reactions which occur in the living systems or within the cell. Okay. nomenclature of the enzyme. How can we name the enzymes? This can be conventional nomenclature and systematic nomenclature. Conventional nomenclature will say, based on these enzymes has three systems of their nomenclature: empirical and broad sense naming. For example, by adding arbitrary suffixes, such as, for example, adding S. For urea, adding S, urease. For lipids, adding S, lipase. For protein, adding S, protease. Systemic nomenclature, based on reactions they catalyze, enzymes are classified into 6 classes by enzyme commission, based on the reactions they catalyze. The class 1 enzyme is oxidoreductase. Oxidoreductase. This catalyzes the oxidation and reduction reaction or redox reaction. The class 2 enzyme is transferase. This catalyzes the transfer of the group from one substrate to another substrate. The third class enzyme is hydrolase. This catalyzes the hydrolysis reaction by using water. The fourth class enzyme is lyase enzyme. This enzyme splits molecules. It does not use water. Also can form, can synthesize molecules, but it does not need ATP. The fifth class is isomerase. This isomerase participated or involved in isomerization of the substrate. For example, mutase, isomerase, epimerase. These are the enzymes of the fifth class of the enzyme commission. The sixth class enzyme is called ligase. This enzyme, as its name indicates, it ligates the molecules or joins two molecules by using ATP, right? This is what differentiates the ligase enzyme from the lyase. Lyase does not use ATP to ligate molecules. Okay, what is the difference between enzyme synthase and synthetase? Enzyme synthase, this is the enzyme which do not require ATP directly, belong to classes other than ligases. The rest classes of the enzyme, class 1 up to class 5, are categorized under this enzyme. For example, glycogen synthase. These are the enzyme which ligate or which binds to molecules without using ATP directly. The enzyme synthetase is the enzyme which forms the macromolecules by using ATP. This belongs to the ligase or class 6 enzyme. For example, glutamine synthetase, arginosuccinate synthetase, and carbamoyl phosphate synthase. Each enzyme is characterized by an enzyme called number, which has 4 digits. The first digit indicates that it is the main group or main class of the enzyme. The second indicates the type of group which is involved in the reaction. What type of the group is involved in the chemical reaction which is occurring. The third denotes more precisely, indicating substrate on which the group acts. Right? The fourth indicates the serial number of the enzyme. Mechanisms of the enzyme action. There are 2 models of the how enzyme bind to the active site. These are lock and key model and induced fit model. Lock and key model says the shape of the substrate and active site of the enzyme are thought to fit each other like key into its lock. The shapes are considered as rigged and fixed permanently, complementary to each other. So the substrate which can fit that enzyme is only the substrate which has the complementary shape which can fit like key into the lock in that enzyme. But when we come to induced fit model, this model says the binding of the substrate changes the conformation of the active site, so that enzyme will be activated and the reaction will go into the product, it says. Just this is the diagrammatical presentation of the key log model. This is enzyme. This is the substrate. As you can see, they have the complementary shape. Few products are formed from key and log model. Okay. This is the diagrammatical presentation of the induced fit model. As you can see, the shape of the enzyme active site and the shape of the substrate are not complementary. So this substrate comes and bind to the enzyme, so it changes, it forms the enzyme substrate complex. And as you can see here, this is the original enzyme, this is enzyme substrate complex. The shape of this and this are not complementary, but in the product, in the enzyme substrate complex, as you can see, the binding of the substrate changes the conformation of the active site. So what will be okay? The enzyme active site forms complementary shape to the substrate after what? After binding. So as you can see, more products will be produced. This is substrate, this is enzyme. They are not complementary. When it binds, it changes what? The conformation of the active site. So products are more. Okay. How do enzymes increase the rate of reaction? Enzymes increase rate of reaction by lowering activation energy of the transition state. Activation energy is the energy difference between the ground state and transition state that occurs during formation of the product. This means the minimum amount of the energy which is required for the chemical reaction to start, right? The transition state means the top of energy hill which decay the substrate or product, which is decay to the substrate or product state is equally probable. Okay, as you can see, this one is without using enzyme. This reaction is by using enzyme. As you can see, this is activation energy. Activation energy. The reaction which is catalyzed by using enzyme is, without using enzyme, is uses high activation energy. That means it is not fast. It uses many hours or even it may be days. But when we come to the enzyme catalyzed using enzyme, the activation energy, as you can see, it is lowered. Okay, without enzyme, this is lactose, high activation energy. Can you see? This is hill. This is too small. The activation energy is low. It does not use high activation energy.